a few. Okay, we just wait just 1 or 2 minutes, just the people to be able to connect and enter the Zoom room. Okay. All right. Yeah, just coming back in one. Okay, so there is still a lot of people in the waiting room. Sorry, Fanny, just a regular few minutes to people to be able to join.
That's fine, no problem.
And in 2 minutes we'll start. Okay. Checking. All right. Is it fine? Can we start, Fanny? Is it fine for you?
Yeah. Yeah,
all right.
That's okay.
So, hello everybody. Thank you for coming to Fanny's talk in NannoTalks. So, Fanny Monteiro is our invited presenter today. Fanny is working for the past 10 years in the University of Bristol. She's now senior lecturer. She has done a PhD before that at the MIT, in US. And before that, she did her master and undergraduate in France because she's born as a French citizen. Fanny's modeler. She's not a paleontologist per se or micropaleontologist. She's a modeler. She's been working on modern and paleo, and she's been working on nitrogen oxygen cycles and a lot on oceanic ecosystems with planktons. So that's why we invited her today to have a different view from a previous talk we had on her view based on modeling. And she's going to present today on that aspect. So thank you very much, Fanny, for coming today. And I give you the end now. Thanks.
Thanks, Baptiste. And also I'd like to thank the group for inviting me to present my work on modeling coccolithophore. So my official title is Revealing the Ecological Impact of Coccolithophore Diversity through Mechanistic and Statistical Modeling. And my informal title is My Journey into the Modeling of Coccolithophore Diversity. I've worked a lot for this project with my PhD student, Yus Dovry, who I'd like to thank today, and with the person who introduced me to statistical modeling, which is a new approach for modeling coccolithophores. And also being a modeler, I work closely with biologists. So I'd like to thank my collaborators, Alex Poulton, Colin Brownlee, and Glenn Wheeler for providing me all the information about coccolithophore ecology. So as you know, coccolithophore are a really important organism for the climate system. They contribute to the organic pump and also to the inorganic calcium carbonate pump. They have a beautiful, diverse morphology and really well described through the taxonomy, and also they're really good fossil records. So despite all that, we still have big unknowns related to coccolithophores in relation to the role of their diversity, in relation to the role and also how classification is actually proceeding, and also the role of mixotrophy and life history. So today my talk, I'm going to focus on two of those aspects, the diversity and classification of coccolithophores. So I've taken this example by Patricia Ziveri, who also is actually here today. So hi, Patricia. She published this work back in 2007, showing actually the role of diversity on calcium carbonate exports. So her data here is taken from 12 different locations, different parts in the ocean, and looking at the average daily flux of calcium carbonate at about 1,000 meter depth. And what the data shows is that different species contribute to calcium carbonate export, including Geocapsa oceanica, H. carteri, Calcidicus leptoporus, and Emiliana axili being the most dominant ones. But current studies focus mostly on Emiliana axili, but it's not even the main producer or exporter of calcium carbonate. So there is a bias towards Emiliana axili that doesn't include the other dominant species. So that drives me to three research questions. The first one is, what are the critical coccolithophore species or groups? My second question, what is their contribution to the global carbon cycle? And finally, my third question, how did or will these critical coccolithophore species and their contribution change with paleo or future climate? So being a modeler, I use different modeling techniques to try to answer those questions. And that includes mechanistic modeling, which is the model of representing different processes, so the interactions between the organism with their environment, and try to make predictions. And also statistical modeling that use observations to find relationships between the organism and the environment. So models can be very useful. In particular, they are useful to fill the gaps of limited data. So this is the observations compilation by O'Brien et al. 2013 of the coccolithophore biomass. And you see that we have a good coverage for the North Atlantic along the AMT in the Atlantic Meridional Transect here, and south of Japan. And there's big parts of the Pacific Ocean, Southern Ocean, South Atlantic, South Indian, where we have very few data. So modeling is useful to, if you want, get global estimates where we fill the gaps. They're also useful for paleo and future predictions, where we have even less data or even no data. And models can be used also to understand the mechanism behind or test hypotheses when we have complex systems like the marine ecosystem, where you have thousands of organisms interacting with each other in a very complex dynamic ocean current flow. So that takes you to the outline of my talk. I will first give you an overview of the modeling of the marine plankton ecosystem, because I assume a lot of you are not too familiar with that, so I thought it would be useful to talk it through. And then I'll present to you the application of a new modeling method for marine ecosystem called trade-based approach, the application to that approach to modeling coccolithophores. And then finally, another new modeling approach for coccolithophore using statistical methods to model the distribution of species, called species distribution modeling. So I will start with the overview on the modeling of marine plankton ecosystems. So how do we model marine ecosystem? And that's the mechanistic approach, where we have all the processes. So first we have an ocean model that represents the physical state of the ocean for the ocean currents, and also a temperature, salinity, light, mixed index, etc. And also to that model, we have some biochemical traces like nutrients, oxygen, pH, anything that we need for the ecosystem. And to that model, we combine an ecosystem model. And the two together, you end up with a distribution of different organisms that you have in your ecosystem. And that tells you about the global estimates, some prediction for the paleo and the future, test hypotheses, or understand the mechanism behind this distribution or a specific response to climate change. So going back in time, the first marine ecosystem model was developed by Riley 1946 with this NPZ model. where the model represents three pools. The first one, N for nutrients, the second one, P for phytoplankton, and the third one, Z for zooplankton. So this model actually was applied for a very specific location and was able to represent the observation. So it's a pretty good first attempt at modeling marine ecosystem. But obviously, when you go bigger, you need to include more diversity. So later on, and that's been kind of the standard model until the late 80s, people to the NPZ model, they added the detritus pool, so called NPZD model. And there they divided the dissolved organic carbon and the particulate organic carbon pools. So that in this model, we were also able to represent the, if you want, the microbial bacterial activity, and also the sinking of the particles into the deep ocean, which is crucial for the biological pump. So those models were kind of like the standards for marine ecosystem modeling until the 80s. And then, as I said, when we start looking at the bigger picture, those models were not able to capture all the different, subtlety from one place to another. So a modeler is starting to include different groups of plankton that they call plankton functional types, PFT. And so they're starting adding one coccolithophore in their model, one diatom, one action fixes, etc. And also with that, they also diversify the biochemistry, adding silicon cycle or iron cycle, etc. So those models really improve the global representation of the biological pump. And also on average, and as we still use those models a lot, we have between 5 to 15 plankton types in those models. What they tend to be defined is based on culture species. So to add a coccolithophore, we take a species that we grow in the lab and look how fast they grow with different light environment, different nutrient environment, different temperature, and then those relationships then are put into the model. The problem with this approach is that we can culture only, very few amounts, about 1% of the species in the lab of the species that live in the real ocean. And those culture species are not necessarily representative of past ecosystem or future ecosystem. So an alternative approach that's been developed since 2007, it's the trait-based ecosystem modeling. That's the one I've been using for the last 15 years. And instead of modeling a specific species, we actually model critical traits. So what is a trait? I'll explain that further on, but an example is, for instance, size. Size is a critical trait of ecosystems. And in addition, we model their trade-off, which I call ecological rules. So here I'm giving an example for this trait-based ecosystem model for the size, where instead of having different functional types of phytoplankton and zooplankton, we have different size classes going for large cells to really small cells. And in addition, we have those trade-off, which are the relationship of the trait here, the size, with critical processes of the organism. So for instance, we know that the nutrient affinity decreases with cell size. So smaller cells are much better at taking up low nutrients than larger cells. We know also there is an optimum in the maximum photosynthetic rate of phytoplankton at about picosize. So the model has this kind of bell shape for the to represent maximum photosynthetic rate. We know that ingestions for the grazers decrease with size, etc., etc. So we have all those relationships that determine the processes of the organism as a function of size. And because size is a master trait for ecology, the model does a pretty good job at capturing the nutrient field. So this is the surface nitrate in the model on the right compared to observations, and as well as chlorophyll A, which is a tracer for primary production. So model compared with satellite data. So that model was developed by Ward et al. 2012-2014. And so to go a bit more in depth with the trait-based approach, there's been a review by Thomas Kjorber in 2018. I really recommend that paper if you want to know more about this approach, where he says that this trait-based approach was really developed for many years now for land ecology, but it's coming on in the last 15 years or so for marine ecology. And he claims it's a very promising tool, And the way he defined the trait ecological approaches is by considering interacting individuals characterized by a few essential traits that are interrelated through trade-off. So there's the key things between defining the key traits and a group of species we're going to have, similar traits, so they will behave similarly, and also the importance of trade-off, which I'm going to explain in the next slides. So the advantage of that approach is that it allows us to represent non-culture species, because if they have similar traits to the culture ones, then you can have them all together. And the ideal to represent diversity and also paleoclimate, because if you define your traits and trade-off fundamentally enough, then you should be able to include species that are not, don't live anymore in the ocean. I'll explain that further. The issue with that approach is we really need to know what are the critical traits and trade-off, and that's not always easy. So what are the traits? For phytoplankton, Lichman and Klaus-Meyer did a review in 2008 where they define morphological traits. So I talk about size, so that's a really important trait for plankton. Also the shape of the cell, or if they form colonies. Then they define the physiological traits. So that includes if the organism do photosynthesis, which type of nutrients they require, if they do nitrogen fixation or mixotrophy. Then the behavioral traits for mixotrophy and motility. And finally, the life history traits, sexual reproduction or resting stages. So then I talk about trade-off. I said each trait is associated with a trade-off, which I call my ecological rules. And it's the idea that a trait has a benefit. So for instance, classification could be to protect against grazers, but there's a cost associated with that benefit. So every time you have a trait that evolves, it's always to the detriment to another trait. And the reason behind is because we have a finite amount of resources. So imagine the situation if there was no cost. So this cross here is an organism that developed a trait which has really high benefit to no cost to another trait. Well, that organism will be super competitive and become a super organism. But in a modern ocean, at least, we in a situation where we have a lot of diversity, so we know there must be a cost associated with a benefit. So that's why we have trade-off, and trade-off generate diversity. So we applied this trait-based approach to plankton ecosystem modeling, and the main model is called the Darwin model. And the way it works is we populate an initial population with 100 to 1,000 different types of plankton, and they are defined by different traits and associated trade-off. So they could have different size, they could have different nutrient uptake, they could be mixotroph or not, etc., etc. And the more traits you add, the more diverse your ecosystem will be in the model. And then this initial population is put into an ocean model with represent all the ocean currents and the light, nutrients, fields. And very quickly, the environment select for the fittest organism. So you end up with an emergent population, which is diverse, and also which is selected by the environment. So you have a real link with what the environmental conditions are. And that's quite a nice approach if you think about, paleoclimate, where the environment was different. You would end up with a different population to the one of today. So it's a really flexible approach if you want to start thinking about paleoclimate or even future. So this approach of the Darwin model has been very successful to represent different plankton groups in the modern ocean, including cyanobacteria, diatoms, nitrogen fixers, mixotrophs, and foraminifera. But what about coccolithophores? So that's going to my second part of my talk, where I'm talking about the application of the trait-based approach to coccolithophore modeling. So in terms of traits, we have quite a good sense about what coccolithophore traits are. So we know their size, we know the cocosphere, coccoliths morphology really well. We know they don't form colonies. I did a classification here as a trait being the main trait of coccolithophore. There's some unknowns about the environmental control of coccolithophore that would be good to know more. We know they do photosynthesis, we know their nutrient requirements. Maybe there's still some unknown about, which species need the silicon or not. There's some debate about nitrogen fixation. I don't know how many of you know about it, but Bristol is found to be doing a symbiotic relationship with the nitrogen fixers called UCYNA. So there is this potential for nitrogen fixation and coccolithophores, which I find very interesting. Then there's some assumption that the deep water species of coccolithophores probably live deep down there because they do mixotrophy. We assume that most species are motile, and then we know they have two life stages, haploid, diploid, so asexual and sexual reproduction. So we have a quite broad understanding about what coccolithore traits are. What about that trade-off? Well, this is where things start to be complicated for modeling coccolithophore in a trait-based approach. We have quite limited knowledge of coccolithophore trade-off. And I realized that as soon as I wanted to model coccolithophore, asking coccolithophore experts, so why do they calcify? And they were like, well, we don't really know. So that brought me to this review back in 2016, focusing on calcification as being the main coccolithophore traits. What are their costs and the benefit of calcification in coccolithophores? So in this review, we first look at the cost, and we found that calcification in coccolithophore is associated with really high energetic costs. About 30% of photosynthetic costs, and it's mostly related to the transport of calcium ion and bicarbonate ion into the cell. We did estimates for two species, Emiliana exili and Calcilicus pelagicus, and we found that the costs vary among species and also vary with the environment. Depending on the pH, you will have smaller or bigger costs. So with such a high cost, 30%, there must be benefits. Otherwise, the organism will not bother. We also look at other potential costs and found that there is a sinking cost. So, the fact calcium carbonate is really dense make the cell sink into the water column, but only for large cells, you have that effect, and only in low turbulent environment. So that's kind of a very specific cost cases. And also what's interesting about calcification, they're associated with no nutrient costs, which is benefit compared to the diatoms that build a first tool made of opal, so they rely on silicic acid. So then we looked at the benefit of calcification, and what we found that they are mostly hypothetical. We identified 3 different types of benefit. One is associated with accelerated photosynthesis, one to photodamage protection, and one to armor protection. And the lab and in situ studies didn't have any conclusive evidence except for protection against protection against UV light, providing like a sunshade, the coccoliths being like a sunshade for this UV light. And also one paper had evidence that the coccolithophores reduced the digestive rate of their grazers. So there is potentially a grazing protection there. All of this data are relying on Emiliana exili, or most of them, I would say. So suggestion is UV sunshade and grazing, and the other one are hypothetical. Looking at the geological record, there is also the evidence that coccolithophore evolved almost at the same time as dinoflagellates, which build organic carbon armor. So maybe that's also suggests, two organisms with kind of looking with an armor could be that the grazers evolved at that time really made that phytoplankton had to find ways around protecting themselves from high grazing pressures. So very hypothetical for coccolithophores. So because we didn't really know the benefits, then what we did in this study is to test the different type of cost and benefit in the Darwin model, in this trait-based model. And the way we did it, we just assumed coccolithophore as a whole group, and then we tested the benefit individually. So in this diagram, each cross is a different experiment, which has a different set for one benefit, for different levels of benefit and different energetic cost. And so we tested 50 different combinations of cost and benefits. So for instance, the cross here was a high benefit for light protection, highlight protection, and with a low cost. And then this experiment here was a high benefit for grazing protection and a high energetic cost. And then we score the model results with observation of total biomass of coccolithophore and diatom along the Atlantic meridional transect, which got a lot of observation and also crosses kind of the key provinces of the ocean. And then we're able to find which experiments was realistic and which were not. So based on this testing experiment of the trade-off, we also find high energy costs between 10 to 50% of photosynthetic rate. And we found that each benefit resulted in a different cost. So depending which benefit, you require a higher cost or a lower cost, but also those benefits had a specific niche. So for instance, grazing protection favor coccolithophore in high latitudes and upwelling region, whilst the light uptake really favor the coccolithophore to grow in a equatorial region and region with really strong currents. The other important result we find doing that testing is that we didn't have any single benefit that could reproduce the distribution of coccolithophore along the EMT. So what we suggested is that there's no single benefit for calcification, but probably multiple benefit of calcification. Either different species are going to have different benefit, or actually within one species you could have multiple benefit of calcification. So based on that, we weren't able to model coccolithophore yet with trait-based approach. And I wasn't able to answer my two last questions about their contribution to the global carbon cycle and how they evolve, if you want, with climate change. So one issue is that we don't know the benefits very well, and also we assume, one group of coccolithophore, maybe different groups have different benefits. So to stop being stuck, I had to find a different way to move forward with the coccolithophores. And that's actually going back to my first question, which is what are the critical coccolithophore species or groups? And so once we identify that, I think then we will be able to go back to the trait-based approach to look at the different benefits for these different groups. So to identify the critical groups of coccolithophore, I'm moving to statistical modeling. So this is the third part of my talk, species distribution modeling, which is a statistical method to represent the distribution of individual species. And this is thanks to the work of my PhD student, Joost de Vries. And what we did the first application of a species distribution model to coccolithophores. So how does that work? First, you take species data. So here, for instance, the biomass of coccolithophore from the Maridat dataset, and then combine it with environmental data, so observations for temperature, salinity, oxygen, nutrients, etc., any environmental data that you want. You then run a species distribution model algorithm, which then tell you the relationship between the biomass or the abundance and the environment. And then based on those relationship is able to fill the gaps. And then you end up with a global distribution prediction of your different species of coccolithophores. So the method we use is a machine learning technique called random forest. It's very powerful. It allows for non-linear relationship between the environment and the biomass, and really to minimize the error between the model and the data. And as I said, this is the first time we've done such a thing for coccolithophores. So it's a very promising tool. The data we use first is a compilation that Joost did in his 2021 paper that came out last year, compiling electron microscopy abundance data. And the idea is we wanted high resolution microscopy to be able to distinguish for all of the species. So we included the 160 most common species and 46 studies equivalent to 3,000 samples. So it looks a bit like the Maridat, but there's a bit less. But it's got high resolution data. So in green is the AMT, in blue is BATS, in red is the Adriatic time series, and then in black and gray is the rest of the data. For the environmental data, we we included temperature, nitrate, silicon, or silicic acid, oxygen, pH, DIC from the World Ocean Atlas, and then light as power, optical depth from the satellite MODIS data. And then we also included iron, but the observation is too sparse, so then we rely on the 3D ocean model called MIT-GCM. And then both data sets were gridded on a 1 degree horizontal grid and had standardized depth levels. So this is a result of estimating the distribution of our 140 species. Here I'm showing the distribution in abundance for Emiliana exili here on the top right, and then of the total population of coccolithophores. So the 140 species all combined together. So what we see is that Emiliana exili has, this high abundance in the calcium carbonate belt in the Southern Ocean, and then high values in the North Pacific and North Atlantic. But then what we did is convert this abundance in biomass, which really transform our estimates for Emiliana exilia. Instead of being 80% of the abundance, Emiliana exilia account for 30% of the biomass in organic carbon biomass. And that's because Emiliana exili is a really small coccolithophore, so actually really reduce their contribution. And that's really important. Biomass is more relevant for climate than the abundance, so it's always important to do the conversion. But what we see looking at the total biomass is that there's really high biomass in the low latitudes. So low latitudes are very important for coccolithophores. Picture that you don't see as much in the abundance data. So there must be other species. So now I'm showing you the top species for abundance and biomass. So for the abundance, you see, Emiliana exili in light blue kind of dominate everywhere, it's the 80%. But then in terms of biomass, Emiliana exili dominates only in the high latitudes and subpolar gyres. For the low latitudes, it's actually in orange, Ambulosphera irregularis, and in light purple, Ambulosphera tenuis, which dominate. And then interestingly, in equatorial subtropical region of the Atlantic is a lot more diverse population. So now I'm showing you the total biomass of the top 15 species of coccolithophores based on this model. And so Emiliana exili account for 30% of the total biomass. And then it's Ambulosphera irregularis with 20% of the total biomass, and then Ambulosphera tenuis for 50%, and UC Bogore equivalent to 10%. So on the first 4 species, 3 of them are umbiliform. So the umbiliform are really important in terms of the biomass. They account for about 45% of the global biomass of coccolithophores. Our next step is also to make the inorganic carbon estimates. So do a conversion using PICPOC data, and then we should be able to get some global estimates as well, which would be interesting. So to conclude, I give you an overview about how to model marine ecosystem, and I hope I show you the potential of trade-based approaches to model non-culture species, diversity, and paleo ecosystems. And then I show you the application of this approach for coccolithophores. And I, as you saw, it's still a challenging task to do. And the main reason is because we have limited knowledge on the benefit of being a coccolithophore, but also what are the main groups of coccolithophore we need to include diversity. I'll also show you that we can use a Darwin model to test for the trade-off. I think this is a very good approach, and we find that there was a high energetic cost still with classification, and that's multiple benefits of classification. And then finally, I'll show you the first application of species distribution modeling to coccolithophore, where we estimate Emiliana exili to be the most dominant species in terms of abundance, contributed to 80%. But then when you convert that into biomass, Emiliana exili still important, but it's not the dominant species anymore. It accounts for 30% of the global coccolithophore biomass. And the other really important player, the umbiliform, which dominates in the low latitude. So those groups are really important to study as well for the modern. So I wanted to finish by giving you a bigger picture of why am I trying to model coccolithophore. And it's this idea that I'm really interested in looking at the interaction between marine ecosystem and climate. And for that, I would like to include the main groups of plankton in paleoclimate modeling. And just to give you a bit of perspective about paleoclimate modeling, so most paleoclimate models are only physical models, which we call Earth system models, or where we represent the dynamic of the atmosphere of the ocean and look at the coupling with different climates or paleogeographies. Many of those climates do have some ocean-based chemistry, so that people start doing the coupling with the carbon cycle or the nutrient cycles, but only a few have got an ecosystem, and it's only some that has the plankton functional type ecosystem models, which is based on modern culture species. And it's only in my group that we developed the first paleoclimate model with a trait-based ecosystem model that we think is the relevant way to look at paleoclimate. So we did that back in 2018, where we combined the size structure Darwin model, so the world model, into an Earth system model called CGINI. And we applied that model for the Eocene, so that's Wilson et al. In 2018, which looked at the change of plankton size during a warm event of the early Eocene. So we're starting to do that, basically. That's what I want to say. And at the same time with my group, I had a PhD student, Maria Guratu, who developed foraminifera, non-spinous minifera in that model, ecogeny. So that's published. And now with Danny Schmidt and Jamie Wilson, we supervise our PhD student called Rui Ying, who is now adding spinous symbiotic foraminifera into ecogeny. I myself working on adding nitrogen fixers into ecogeny to look at oceanic anostic events, because with a biogeochemical model, we saw that OAEs are favoring nitrogen fixation and probably influence the evolution of nitrogen fixers. So there's a lot of work going on there. And then at the same time, with Daniel Conley and his postdoc, Aaron Naibu Backwell, we're adding diatoms into ecogeny to look at the evolution of diatoms during the Cretaceous. So the idea is eventually we'll have all the main, functional groups of phytoplankton in paleoclimate models, and we should be able to do some interactions with ecosystem and climate. And I really want the coccolithophore in there. So talking to paleontologists and observationalists, which data would I need to model coccolithophores in trait-based model? Well, I need data for a more diverse group of coccolithophores to start with, no doubt. We have too many data. We have lots of data for Emiliana exili. It'd be good to have, include the other groups. We need more conclusive evidence for the benefit of being a coccolithophore. I haven't touched on that too much, but it's evident that we need more understanding on the environmental control on photosynthesis and calcification for the main coccolithophore species, so that we can really see the influence of carbonate chemistry or the environment on, the peak-POC ratio, which is key for climate. I'm really interested in including mixotrophy and nitrogen fixation in relation to coccolithophores, but at this point, we haven't identified the species that do it. So having that information would be very useful. And then for the paleo application, I need data to compare the model with. So it'd be great to have historical data of the main traits of the coccolithophore. So I know there is size, which is more or less available. So here I took an example by Hannes et al. 2012, showing the evolution of coccolith size, very good for the cell size as well. And also do the same for the peak-POC ratio and the different morphology, etc., etc. So I hope I inspired you to look into the modeling of coccolithophores and the trait-based ecology. And I stop my talk here and take any questions. Thank you.
Thank you, Fanny. Thank you. It was a brilliant talk. Thank you for the wish list. We should have invited you in late December, I guess, for Christmas wish list. But I guess
well, maybe next Christmas.
Exactly. So if anybody has a question, you can ask the question. Just raise the hand, and I will give you the possibility to ask yourself the question, or you can write the question in the discussion tab.
Baptiste, should I stop sharing? What's the best?
No, you can stop sharing, but if you want to keep it to answer a question, well, I guess you may need it. I don't know, depending on if anything, you can stop and share again if you need it.
Okay, I'll keep it for now.
All right. So, Jeremy, I'm pretty sure you have a question. Jeremy, your microphone is off.
Unmute. Hi, can you hear me?
Yeah.
Yeah, nice, lovely talk. One of the thoughts which occurs to me is, particularly when doing looking at the umbelliform ones, is how important is growth rate? Because I have a suspicion that maybe Umbellispira is growing much slower than EHUX, and so if you simply take the standing stock biomass, it may not be representative of its biological importance.
I see your point. At the same time, the standing stock is almost the same as Emiliana actually. So in terms of carbon, they're as relevant. And yeah, well, that would be interesting when we do the tray-based modeling. We will have them, you know, with their growth rate, and then we could really estimate the export directly. But I don't know. I think, yeah, it's debatable. I think this should be as important for exports as their standing stock.
Yeah, I think it's a big unknown which we really need to, in order to get the model, is to know what those differential growth rates are, because certainly it's one of these anomalies that in the plankton we have these enormous abundance of Umbellispira, but we don't see that in the sediment traps to anything anything like the same degree. And so, as I say, it seemed to me that one of the most likely interpretations is that it's a much lower growth rate. So I think you probably need to add it to your wish list.
Yeah. And also, what about, because it's the preservation in the water column, right? When the umbelliform maybe is not as robust as Emiliana actually. I don't know, I'm asking.
Yeah, it doesn't. Well, I guess Luke would be another good person to ask that. But my feeling is that just preservation can't explain how fast we seem to the abundance drops off. I strongly suspect it may just be they're growing slower as well. And it would also sort of help explain why they're difficult to get in culture, because generally the slow growing ones are the ones which are hard to get in culture.
Yeah, definitely.
Okay, Luc was raising his hand, so I give you.
Yeah, thank you, Fanny, for this beautiful talk. Very interesting, and I really liked it. Do you hear me?
Yeah, very well. Yeah. Hi, Luc.
Good. The question was very similar to Jeremy, and he already asked one part of my question. And the second part was the depth for the umbelliforms and over coccolithophores, because usually they are very at shallow depth in the photic zone. Instead, Emiliana Huxley I and many others are found deeper in low latitude. So how did you manage with the depth?
Yeah, so I'll show you actually the result with depth. So that's the biomass estimates. So the model represents, so I'll show you the integrated vertical biomass or abundance in the previous plot. But we have also the vertical representation. So here you have total biomass of coccolithophore as a function of latitude on the x-axis and depth on the y-axis. Emiliana actually, as you said, is dominant in the surface, but they actually, in the model at least, go quite deep in, the calcium carbonate belt region here. And then the umbiliform, it depends on which type, but tenuis and irregularis are quite surface, subsurface. But the Cibogae here, it seems to be more, at about 50, 100 meter depth. So that group, particular group, seems happier, deeper. And so in terms of the dominant species with depth, so Cibogae is in dark orange here. It's really the dominant one in the model. Whereas the light yellow is the surface one, and that's the Umbiliphora irregularis.
Yeah, but
and then,
this is strange to me. I understand that I really accept this, your diagram here, but usually in the low latitude, we found that at higher depth, there is a higher abundance in terms of quantitative abundance. And therefore, I'm surprised for any type. Usually it goes with the deep chlorophyll maximum where you have the highest abundance. So I'm surprised by the group which is at the surface is dominant in terms of quantity. But maybe I'm wrong.
But that's biomass. I don't know if that helps. I've got also the abundance here. But it's the same.
Biomass.
It would be the same.
The biomass will be higher in the deep chlorophyll maximum. This was my point, in fact. My surprise, not my point. And,
okay. So deep chlorophyll. I could ask, Joost is here, is the one that worked on that. Joost, do you have any idea about Luke's question about the deep chlorophyll maximum in the low latitudes? Should be the way we find the highest abundance or biomass. I think he was here. Yeah.
Yeah, I'm here. Sorry. It's a good question. I'm not entirely sure, to be honest. Something we have to look into a bit more. One thing also looking at optical depth rather than on actual depth as plotted here. Yeah.
Okay. Well, thanks, Luke. Something to look at.
Thank you very much.
I haven't said this is still very new results, by the way. So we're still looking into the data. There's quite a lot of things to explore.
It's beautiful anyway. Thank you.
Thanks, Luke.
Thanks. Patricia was raising her hand, so I give you the hand then.
Hello, Fanny. Thanks a lot for the presentation. It's really great. The highlighting the complexity of the coccolithophore biodiversity. So my question actually is a follow-up of Jeremy and Luke. And I agree about actually the comments of Luke. I was also surprised about the issue with the deep chlorophyll maximum and the biomass. But my question was about two things that you didn't cover so much in your talk. One, it was about Emiliano axis blooms and how much they bring in terms of biomass. You don't see any sign of Emiliano axis blooms in your global plot. And the other question is about the seasonal correction of your biomass, if it's been done or not. That it can be, of course, quite different when you're moving up in the latitude from low to high latitude.
Yeah, so it's a really good question. So I'm showing maybe this one. What I'm showing here is the annual average. So that's probably why we don't see the blooms. But then when you go by seasons, we do have blooms. And again, I could go to Joost to say, as he looked at that in more details. So the blooms should be there. And then in terms of correction for the seasonal, so you're saying we sample mostly in the summer months or, spring, summer months. Is that your question? Is there a bias in a model related to that?
Or if you made some sort of a correction to get the seasonality.
So
done.
I know we had to add some mask for the winter months in the high latitudes where there's no one that goes sample, but we know there shouldn't be any coccolithophores growing there. So we bluntly add a mask saying there's nothing there. So the model was not calculating. And before I did the mask, we were just having coccolithophores in the winter in the North Atlantic. So we did that correction. I don't know, Joost, do you want to add? Did you do anything else?
No, just that correction. No, we don't extrapolate beyond kind of what we sample. And to add to the seasonality, we do see seasonal trends in terms of kind of Southern Ocean blooms and such, and also abundance moving up and down with the seasons.
Yeah, Rachel, I hope that answered your question.
Okay, thanks. Yeah.
Thank you. Any other question? Let me check on YouTube at the same time. Wait. Oh no. Yeah. Okay. There were 7 people on YouTube. I mean 6 because I'm also on YouTube. But there were no questions. Okay. So I do have questions, if I may. I just wonder, because you at the very end, and I'm pretty know this question is a bit very early in your research program, but you said you wanted to go also to paleo, and you were talking about oceanic anoxic events. I know that you've been working on that since your PhD or something like that, if I remember. But if you go to, for instance, I know quite well the Tuarcian oceanic anoxic event, and the coccolith world is completely different. The foraminiferous world is completely different. How will you use a model based on observation from modern in a world that is completely different? There is, I mean, there is almost no plantic foraminifera. Actually, there is no plantic foraminifera. The coccolithophore seems to have completely different way of living in the oceans based on a few studies from Emanuela Mattioli's. So how will you base your observation on today and also try not to be too much into those observations?
Yeah, it's definitely a challenge, right? But if you were to look at the traits, so look at those. So for instance, the foraminifera, there is no planktonic foraminifera, they were all benthic. So that would be, you know, the first thing. So you would need to include the benthic foraminifera because yeah, at the moment it's only the planktonic ones. Then for the coccolithophore, again, you will need to look at the fossil record and understand, you know, what are the specific traits that characterize that paleo population, and do you have those in a model or not? And maybe there is, you know, an example in a modern, even if it's not a dominant group, but maybe you could test a little bit if the model is doing a good job or not for that particular trait. Or if it's like, you know, a trait that exists for another group which is dominant in a modern, maybe you could do some correlation there. For instance, nitrogen fixation is a very energetic demanding process. That means that the organism doesn't rely on any inorganic nutrients, any inorganic nitrogen like nitrate or ammonium. Those are quite fundamental principles. So then if I want to have nitrogen fixation with the coccolithophores, I know those, you know, will be applicable. So those would be like the kind of the fundamental trade-off for nitrogen fixation in coccolithophores, for instance. So I would start it this way. Obviously, it's nice to have the fossil records because then we can test the model. That's why I think starting with the fossil species is really key if you want to do the marine paleo climate. The other thing you have to be aware is better than actually do the functional type or the Bajik model who are tuned to modern species and assume that the species haven't evolved at all. That's what we do at the moment with paleoclimate. And I think what the trait-based approach allow at least for, you know, a change in ecosystem structure with paleo. And then it would be super interesting to question, so why were the coccolithophore, the foraminifera so different to today? Is it the environmental condition that, you know, kind of a Darwinian approach view, or is it because there's that kind of a history of evolution that kind of made the foraminifera to be benthic first and then the planktonic foraminifera arrived next? So I think, you know, even if you don't get the right answer, it could tell you things that's happening in the ecosystem.
Thank you very much. Okay. I really agree with you. You need to test a lot of stuff at the end, and we'll give you a bit of the story. Any other question? Otherwise, I have another one that very, very quick, but if there is anybody else having a question. Okay, so just very quick, you say that I understand that you don't have carbonate yet in your results, because you said you wanted to add now work on the peak, if I remember well. Do you how do you estimate the impact on the carbon cycle having adding the peak, because you have calcification that release CO2, but also if you're exporting carbonate to the deep oceans will also influence the carbon cycles? How will you manage this different time different parts of the influence on the carbon cycles? One that is very quick based on calcification, the other one exporting to the deep oceans. And you also have CCD, I guess, that will mess up stuff around. So is it already in a part of CGNEs or something that you need to develop to?
Yeah, so CGD does have a very good calcium carbonate cycle. It already does the CCD and carbonate export, and even does burial of calcium carbonate, and compare fairly well with observations for modern and paleo as well. The challenge is, when we tested it for coccolithophores, is that coccolithophores is not the only calcifier. So we will be able to have calcification in a model that links to environmental conditions, so pH or light or nutrient, etc., which is already a challenge, honestly. But I think we're getting closer from being able to model that. So we could do that and then estimate, how much calcium carbonate is produced from coccolithophores. But we can't link it yet with the carbonate chemistry or the carbon cycle, because we will need to include all the calcifiers in the ocean. So at the moment, if I was to switch on calcification in my model, whatever, even if I got the best distribution of coccolithophores, the alkalinity will go bonkers in the model, because I don't have the foraminifera or the pteropods or, all the calcium carbonate platforms in shallow waters. So you will need to include that as well, so that you don't mess up your carbonate chemistry in the ocean. So that would be quite a challenge to get to that level. But I think if you could already say, look, for future climate, we have ocean acidification events, and that's going to favor that species over other species of coccolithophores, and that will change the bulk calcium carbonate export by as much. I think that would be already quite a big way forward.
Okay, thank you. That's big change then.
Yeah.
Any other question? Checking the different. Okay, so if there is no other question, I guess we can close the talk for today. Thank you, Fanny. It was very, very interesting talk. I really enjoy it. I have obviously many other questions, and I hope I'll ask you in the future about to meet you. And if you're not aware, there will be a conference in Avignon at the beginning of September, the INA meeting, biannual meeting. Obviously, will be very, very interesting having you at this conference. And it's in the Palais des Papes in Avignon, so it would be a very nice venue, obviously. Just a few words on the next NannoTalks. It will be Andy Bowman that will present taxonomy on helioliths. We didn't have any taxonomy yet in the NannoTalk, so we wanted to also invite someone working on that aspect, and it will be in the middle of February. Let me remember. It's the 16th, Wednesday, 16th of February. I don't remember exactly the hours because he's living in central US, so we'll give you the hours by email. And that's it on that aspect. So thank you very much, Fanny. Thank you everybody for coming, and I hope to see you in the next NannoTalks.